Hello to our friends joining us via recording. Today we are talking about lesson number 6, which is the central dogma. And when I use this term central dogma, it sounds pretty scary, but I promise that the central dogma is not as scary as it sounds. All the central dogma is explaining to us how we start with our genetic information and we turn that genetic information into proteins. So our goal today is to talk about the first half of the central dogma. Next time we meet, we'll talk about that second half of the central dogma, how we build our proteins. So here's a picture that's a little bit bigger, a little bit more detailed for you, of showing you what the central dogma is. So when we talk about the central dogma, we start with our genetic information called DNA. Can you remind me in the chat, where is your DNA located in your cell? Where do we normally find DNA inside your cells? Lab review question, right? Get us ready for the midterm. Exactly. Yep, several of us are chiming in the chat. Your DNA is found in the nucleus of your cell. Part of why we have to do the central dogma is because DNA is found in the nucleus, but we build proteins not in the nucleus. There were a couple of different organelles inside your cell that help to build proteins. Can you all remind me? There were 2 different organelles that build proteins. There was the really small ones, and then there was that bigger one. Yeah, so the really small ones, we're getting that into our chat. So the ribosomes are the really small things that make our proteins. There's one other thing as well that helps us to make those proteins. So good guess, Victor, on the Golgi apparatus. The Golgi apparatus receives those proteins and packages them. Yeah, Margarita's right. The other organelle that builds proteins is the rough ER. So the rough ER builds proteins, the ribosomes build proteins, and then they send those proteins to the Golgi apparatus, who packages them up, makes them look good, and sends them throughout the cell. When we talk about the ribosomes and we talk about the rough ER, neither of those things that you told me build proteins, neither of those live inside the nucleus, but that's where the DNA is found. So the reason we have to do this big, long central dogma thing is because your DNA is found inside the nucleus of your cell, but I don't build my proteins there. By the way, down here at the bottom, they're labeling this amino acid chain a polypeptide. That's another name for proteins. We can call them polypeptides. And that's based on the name of these special chemical bonds that I see inside my proteins. Does anybody remember? And again, use this name to help us out. What's that special kind of chemical bond that only proteins have? What's that special protein bond? I told you anytime you see this word, we are thinking proteins. Exactly. Yep. Several of us either remembered it or we saw it here in the name of our protein. A polypeptide is a molecule that has lots of peptide bonds. And peptide bonds are the specific kind of chemical bonds that we use to put together amino acids to build us proteins. Bernice's saying she's having some issues hearing me. Is that across the board? Or is everyone else coming in good? Okay, so what I might recommend, Bernice, is maybe check your speakers, but if you're still having trouble, maybe go out and come back in. That might help you to hear a little bit better. Yeah, most of us can hear me, which is perfect. So if you're still having trouble, just try going out and come back in. That might fix it for you. All right, so when we talk about the central dogma, let's start with the discussion of the genetic information in your cell. We're going to start with this stuff here on the left called DNA. So DNA exists as what we call a double helix, meaning there are 2 strands to your DNA molecules. When we talk about these 2 strands of our DNA molecules, there's a word that we use to describe them when we're talking about them relative to each other. I'm going to type it here in the chat. The word to describe the two strands of our DNA is called complementary. It's really important that we know what complementary means, because that's gonna be, I promise you, will be on quizzes and be on the exam. When I say that the two strands of DNA are complementary, I mean that they are not identical to each other. They don't exactly match. But if I know the sequence of one side of my DNA, I can predict the sequence of the other. Yeah, exactly like Jessica mentioned in the chat. The two sequences of DNA mirror each other. So if you go and look at yourself in the mirror, you don't look identical, you look kind of opposite. Your left side and your right side are opposite of normal. That's the way that it is in DNA strands. So when we look over here at my blue strand of DNA, notice that my blue strand of DNA starts with an A, with an adenine, then it's got a G, guanine. We go down here to a thymine, then we've got a cytosine. That's the sequence of this strand of DNA. My other strand of DNA, its complementary DNA, has kind of the mirror image sequence. So I had an A over here, which means that next door I have a T. A and T always go together. I had a G over here, which means I've got a C over here. G and C always go together. So notice down here how I've got a T in this strand of DNA, which means now this one has an A. And down here I've got a C, which means this one has a G. So when I talk about my strands being complementary, they're not identical to one another, but I can use them to predict each other. Over on the right side of my screen, you see a bunch of chemistry going on. And please understand that I promise you, I will not give you a picture of this chemical and ask you to tell me which of my letters does this represent. I promise I'm not going to ask you that. The reason I included this for you, though, is I want to help you understand why A will only ever pair with T, or why C will only ever pair with G. And it comes down to their chemical structure. So let's look at the chemical structure together to help us understand why these things work this way. Over here on the right, we are seeing the base pair adenine, or A is how you'll always see it abbreviated. When we talk about adenine, notice that it has 1 and 2 places where it builds these hydrogen bonds. For me to build a hydrogen bond, remember that I have to have dipoles. So here I've got a negative dipole. Up here I've got a positive dipole. I have 2 different dipoles that want to interact with another nucleotide. Well, it just so happens that thymine, T, also has 2 dipoles. It has a negative dipole and a positive dipole. Because both of these nucleotides, A and T, have 2 dipoles, they want to interact with each other using these hydrogen bonds. So A and T makes 2 bonds together. That makes it so that they cannot make bonds with cytosine or guanine, with C or G, because C and G both have 1, 2, and 3 dipoles. So cytosine and guanine always want to pair up with 3 hydrogen bonds. Adenine and thymine always want to pair up with 2 hydrogen bonds. Because they have different chemical structures, that's what's going to decide whether or not they can interact with each other. Fun little tangent story for you. Dr. Allis has a favorite nucleotide. Dr. Allis's favorite nucleotide is adenine. Don't judge me. I literally have a cat named Adenine. We call her Addie for short all the time. But she is adenine when she's like on the table cleaning a plate, like licking a plate clean. Adenine, get off the table. Or adenine, move. So Dr. Allis is a total nerd. I have a cat named Adenine. We call her Addie. My little fiery tabby cat is named Addie. So more than you wanted to know about your teacher, but hey, the nerd runs deep, so I had to share. When we are talking about complementary DNA, we can use what we just talked about with A's and T's and G's and C's to help us build the complementary strand. So you can see listed here on the page, and this is in your lecture notes as well, a DNA sequence up here. I've given you the blue line. I want you to predict the red line, or I want you to build the complementary DNA. So let's make a quick list for each other. Every time that I see a T in one DNA strand, what do I put in the other DNA strand? If I've got a T here, what does T like to pair with? Yeah, T likes to pair with A. So I'm going to have an A. I've got an A up here. What does A like to pair with? I'm just flipping it on its head, right? So A likes to pair with T, absolutely. Now I've got a C. C likes to pair with, exactly, G. And G likes to pair with C, the opposite. All right, so I've done the first 4 nucleotides for you. I'm going to give us about 30 or 45 seconds. In your notes, or if you didn't print the notes, grab a scrap of paper really fast. Let's turn this one DNA strand into its complementary DNA. Let's make the other half of the double helix. So I'm going to give you some time to do this here. When you have finished that complementary DNA strand, can you shoot me an emoji in the chat? Once I have a lot of this done, we'll move on. If you're still working, keep working. You're totally fine. We got time. Aranza, I like that you had a light bulb moment. That makes me happy. Lots of love. This is perfect. Ooh, Samantha is excited. Love it. All right. So lots of us have finished this. If you're still working, that's totally fine. But just like we did as we started together, every time we have a T, we're always going to put an A in our complementary DNA. Every time we have an A, we're always going to put a T in the complementary. Every time we have a C, we put a G, and every time we have a G, we put a C. So you will see on the homework assignment, and I am going to ask you to do this on quizzes and exams as well. I'll tell you, here's a DNA sequence. I'll give you one of the strands and say, build me the complementary DNA strand. Build me the other half of the double helix. And remind me again, because I'm too forgetful, when I say that the sequences are complementary, that means they are or are not the same. If we're complementary, we are or are not the same. Exactly. Yes, we're chiming in on the chat. If we're complementary, we're not the same. We're kind of like opposites. We're a mirror to each other. So keep that in mind. We can make complementary DNA to help us build that double helix. What we have talked about so far is DNA. So DNA, like we see in our picture here, it's double-stranded. It has 2 strands to it. But when we're talking about how to get directions from in the nucleus to out of the cell, we're not actually going to use this DNA. Remember, DNA lives in the nucleus. It has to stay in the nucleus to stay safe. We're going to use a molecule called RNA that's only single-stranded, and that can go out of the nucleus and into the cell, where we can see it by the ribosomes, where we can see it with the rough ER, whoever is going to turn it into proteins. So when we're comparing what stores our information to what can take that information other places, a few important things. Number 1, like we mentioned, this has 2 strands versus this having 1 strand. That's the first thing that's different. Number 2, the second thing that's different is the kind of sugar that we find in the backbone of these molecules. So when I talk about the backbone, I'm meaning these ribbons. So notice how in DNA we have 2 ribbons. We have a red one and we have a blue one. In RNA, I have 1 ribbon. It's this beige ribbon that we see over here. When I'm talking about DNA, deoxyribonucleic acid, the kind of sugar that helps me to build those ribbons is something called deoxyribose. Tapping it in the chat for us here. Deoxyribose. So most of its name is right here, right? Deoxyrib, and if you just add a SE, deoxyribose. Here, I'll add that for us so we can see it in here too. Deoxyribose. That's the name of the sugar that we find inside the backbone of DNA, which is how DNA got its name, deoxyribose. RNA doesn't use deoxyribose. RNA just uses ribose sugar. For the most part, whether I'm building with DNA or with RNA, or if I'm building, with deoxyribose or ribose sugar, most of my nucleotides, most of my letters play nice with either sugar. Most of them are cool with that. But there is one letter who does not play nice with ribose sugar. Which of my 4 nucleotides from DNA do I not find in RNA? Who does not play nice with ribose? Exactly. Yep, several of us chiming in the chat. Thymine, or T, does not play nice with ribose sugar. Thymine is like, I am too good for this. I will not attach to ribose sugar. So when I'm looking at RNA, instead of having thymine, RNA uses a different base called uracil. Uracil. All of my other bases are totally the same. They're totally fine. I still have adenine, I still have cytosine, I still have guanine. But instead of having thymine, now I have uracil. So when we're thinking about those comparisons, and I am going to have you build some RNA here in just a moment, some of our relationships are still the same. So if I have a C in one strand in my DNA, when I go to make that RNA, I'm still going to put in a G. Or vice versa. If my DNA has a G, my RNA is going to have a C in it. But things get funky when I have an A in my DNA. If I have an A in my DNA, when I build RNA, remember, thymine is too cool for ribose sugar. So anytime I have an A in the DNA, now my RNA is going to have a uracil. If my DNA has a T in it, that's no big deal. I can put in an A with that. So everybody except thymine, totally cool with ribose sugar. When we build complementary RNA, we have to take that into consideration. We only have uracil to work with. We cannot use thymine, which here's a wink, wink, nudge, nudge for you. If you ever see a sequence and we can't tell if it's DNA or it's RNA, look for the T's or look for the U's. Depending on which letter you have, that's going to tell you if you have DNA or if you have RNA. So keep that in mind when we're distinguishing between those two. When we talk about RNA, ribonucleic acid, with the ribose sugar. There are actually technically 3 kinds of RNA. The one that we're going to focus most on today is this one up here, messenger RNA. When we talk about messenger RNA or mRNA, this is the typical RNA that we were just talking about. So it has one strand. The job of messenger RNA or mRNA is to go from the nucleus into the cytosol so that we can take that message and build a protein. A couple of other kinds of RNA. We're going to care a lot on Wednesday about tRNA. Technically, tRNA is called transfer RNA, but you might want to think of it, especially after Wednesday, as translator RNA. So this is what's going to help us to go from genetic information into amino acids, which is what proteins are built out of. So some of my RNA is messenger RNA, sends messages. Some of my RNA is transfer RNA, helps me to go from nucleic acids into a protein. One other kind of RNA that we're not going to talk about except to tell you that it exists is what we call ribosomal RNA. So there is actually some nucleotides inside your ribosomes as well. We're not going to talk a lot about that, but sometimes I'll mention, for example, the research that I did as a graduate student for my doctorate. You can look at the sequence of ribosomal RNA to figure out the identity of the bacteria that you're working with. So we're not going to spend a lot of time on our RNA, but it can be used in sequencing to figure out who you're working with. So fun sidebar there. When we talk, I'm going to back up here, so whiplash for a moment, with me back to here. When we talk about the central dogma, I mentioned at the beginning of class, we're going to focus on this. We're going to focus on the process of going from DNA into mRNA. That's our goal today, the process of transcription. For us to do transcription, we are going to focus on just a particular part of our DNA, the part that we actually care about. That's what we're going to turn into RNA to help us build the protein that we're looking for. So when you're not expressing your genes, your DNA double helix looks like this all throughout your cell. We have all the directions we could ever need. In the process of transcription, we're going to focus on just the directions we're interested in. So whiplash again, back to our picture. Let's say I am a skin cell. And because I'm a skin cell, I really need to build the protein keratin. Ooh, that looks really terrible. Sorry, guys. I need to build the protein keratin because keratin is going to make me tough or resistant to damage. In my DNA, I have the instructions for building keratin, and I have the instructions for building melanin, and I have the instructions for making your eye color. I have a lot of information, all the information I could ever need. But when I'm a skin cell, I only care about keratin. So the way that I can help my cells help me to build keratin is I'm going to organize that information in a gene. So genes are the set of instructions for particular things I want to build. To help me figure out where those particular directions are, I have to use this thing called a promoter region in front of that gene. So think about promoter regions kind of like a billboard. Now, I'm going to age myself here, but I'm going to ask anyway, is there anyone else who was driving before they had a smartphone? Do I have any other dinosaur friends? Shall we call ourselves? Okay, got a few friends here. Okay, so people who were driving before you had a smartphone, back in the day before Google Maps or Apple Maps or whatever you use, you'll remember that if we wanted to know what was off the highway, we had to watch those billboards because they told us exit here for whatever. So I've told you all, I'm from Oklahoma. I've done a lot of road tripping between Oklahoma and Texas. So if I'm doing the Oklahoma-Texas road trip, the thing that's like the quintessential road trip food for me is a Brahms mint chocolate chip shake. That is what does it for me. So I'm driving down the highway, or I'm driving up the highway, depending which way I'm going, and I'm watching, because I don't have a smartphone, I don't know where Brahms is, but I see a billboard that says exit here for happiness. Not actually, it just says Brahms here, right? But like, that's what it means to me. If I leave here now, I can get that shake. I use that billboard to tell me where the food is that I want, what I'm going to get off the highway for to go get my shake. That shake is like the gene in our discussion here. I want to get that mint chocolate chip shake. That's what I'm after. So I'm going to look for the promoter region, or the billboard, that tells me I'm going to find the mint chocolate chip shake. Or maybe I'm doing one of those like late night trips, and instead of a mint chocolate chip shake, maybe now I need coffee. I'm going to look for a different gene, or I'm going to look for a different location like Starbucks to get me that coffee. I need a different promoter region, or a different billboard, to tell me where Starbucks is. So think about this promoter region like the sign that tells you where you need to get off the highway to go get the food or the drink that you would like to get you to the gene you're interested in. Hey, we've been a little quiet in the chat. Help me out. What billboards would you leave the highway for? What's your road trip food? What makes us leave the highway to go find that gene of interest? I'm a Brahms person. Okay, I gotta vote for Chick-fil-A. My daughter loves the fruit at Chick-fil-A. It's just the fruit. It's nothing else. She loves the fruit. McDonald's. Yep, I got 2 kids that love McDonald's. Buc-ee's. I had a lot of votes for Buc-ee's this morning. Yep. Yes, everyone's saying, ooh, Buc-ee's. Yeah. Okay, so next time you're driving down the road and you see that sign for Buc-ee's, remember, that's just like a promoter region. It's telling you where we would need to exit to go find Buc-ee's, to find the information or the food or the experience, whatever we want to say for Buc-ee's, to find what you want. When we talk about building the protein that we want, when we talk about expressing a gene, that's what it's called. It starts with an enzyme, a protein called RNA polymerase. Now, RNA polymerase, the second part of its name means that it builds something. I promise this is not a trick question. Based on its name, RNA polymerase, what is this going to build? What does RNA polymerase build? And I promise it's not a trick question. So polymerase means I'm building something. The something that I'm building is in my name. RNA polymerase is an enzyme that builds RNA. This is what's going to get the information that I'm looking for, my DNA, that particular gene. It's going to turn it into something new that is RNA. RNA is what can leave the nucleus to go out into my cell to be used to build a protein. So when we're trying to express a gene, when we're getting ready to build a protein, we start with this enzyme, RNA polymerase. You can see it big here in gray. The first thing that has to happen is RNA polymerase has to attach to the promoter region. Remember that billboard that we were talking about? RNA polymerase is looking for the billboard to tell it, hey, here's the gene for keratin. That's what I'm looking for. There's my billboard. RNA polymerase attaches to the promoter region. And when it attaches to the promoter region, the double helix opens up. So we split apart the 2 strands, number 1 and number 2 of my DNA. I attach to it, and then RNA polymerase works its way through that gene. It reads the information in the strand of DNA, and it turns it into a molecule of RNA that we see here. So the process of transcription starts with RNA polymerase attaching to the DNA. Once it's attached to the DNA and the double helix opens, it's able to go through and read that and make complementary RNA to make a copy of that genetic information, but in a single-stranded form, instead of the double-stranded form that DNA is. I want to emphasize something with you before we practice with building RNA. We talked about the promoter region being that billboard that helped us to know it's time to get off the highway, go find your food. But what would happen if that billboard was gone? So Texas and Oklahoma, we're kind of tornado alley, right? We haven't had too many tornadoes here lately, but in Oklahoma, we definitely had more. So let's say that a tornado came through and it took away my billboard for Brahms. And I'm driving down the highway and I don't see the billboard for Brahms and I don't have a smartphone. I don't know to stop. That's the same way it is inside your cells. If the promoter region gets damaged, so maybe we have a mutation in the sequence for the promoter region, or maybe somehow our DNA gets cut and the promoter region is like broken. If that promoter region isn't intact and isn't correct, just like if that billboard's gone, we can't do transcription. We can't find the information we're looking for. We can't turn it into RNA. So the big idea that I'm getting at with you, that promoter region needs to be working for us to be able to do transcription. If I don't have my billboard, I don't know where my information is. I can't copy that information. So promoter region, very important. Important that we don't have that get a mutation or get broken. All right, so we talked about RNA polymerase, attaching to the DNA, reading the DNA, and building complementary RNA. So check this out. Here's the DNA sequence that RNA polymerase is reading. It reads a T, and it says, sweet, I'm going to put in an A. It reads an A, and remember, if I was in DNA, this would go into a T, but RNA doesn't have T. RNA has U. So it puts in a U every time it has an A. Here's that C. Every time there's a C, we still put in a G, and vice versa. There's a G in the DNA, I'm going to put a C into my RNA. So the process of building complementary RNA is just like the process of building complementary DNA, except I have to use that uracil instead of the thymine. So let me pause for a moment. We've got a sequence here that I'm asking you to transcribe, or in other words, I want you to do transcription. Here's the DNA. I want you to build the complementary RNA. I'm going to stop talking. Give us about 45 seconds. Let's build our complementary RNA to this DNA sequence. When you get that complementary RNA made, can you send me an emoji in the chat? Want to get a feel for how many of us have finished transcribing, doing the process of transcription. Awesome. Okay, so it looks like a lot of us have finished that. I'm not going to do it all with us together, but we'll do a few of them together. So I'm going to have you all help me out because you've already done this, right? My DNA sequence has a T. So in my RNA, what am I going to put in at the beginning? DNA has a T, so the RNA is going to have, yep, we're going to have an A. T always goes with A. I've got an A in my DNA. That's problematic because now we're an RNA. What do I have to pair an A with? Yeah, I have to pair an A with a U. Got to put a uracil down here. Absolutely. Now I've got a C. Just like in DNA, C always pairs with G. Exactly. And now I've got that G in my DNA, and that always pairs with a C. Exactly. Hey, here's a wink, wink, nudge, nudge for you. This complementary RNA that you made is actually the exact same sequence as the complementary DNA that you made. It's just that it has uracils now instead of thymines. So if you can make complementary DNA, you can do complementary RNA too. All you got to do is trade out those T's with a U and you're all set. So the process of transcription, turning DNA into mRNA, going from one kind of information molecule into another. There is an analogy in your guided lesson that I want to take a little bit of time to talk through with you, and then I'm going to split us up into our group work times for today. So the analogy that I'm going to chat with you about is the way that Dr. Aulis, little old lady Dr. Aulis, used to have to do her book reports when Dr. Aulis was a kid in elementary school. So do I have any friends here in the class who also had to go to their public library to get references for their book reports? That was totally my elementary school experience in the days before the internet. So let's chat about how this works. So let me grab my laser pointer here. So let's say this is like third grade Dr. Aulis. Third grade Dr. Aulis needs to do a book report. And one of the things that third grade Dr. Aulis is really interested in is mummies. For some reason, I really enjoyed learning about ancient Egypt. So I want to go to the library and I want to get some information about mummies because they're cool to me from a book report. I go to the library and there's this set of books that we see right here. This set of books is called Encyclopedia Britannica, which before the internet was like the sum of all knowledge. So if I want to learn about mummies, I'm going to go to Encyclopedia Britannica and I'm going to find the particular edition of Encyclopedia Britannica that has M in it, that has information about mummies. Now, when I'm doing a book report about mummies, even though I normally am really interested in aliens, that would be up here with A, I'm also really interested in tornadoes, that's down here with T. All I care about is M. I only care about the mummies. So just like when we're inside your cells have DNA that has all of the particular information that you could ever need. Every single gene that you express in your entire body is in your DNA, in every cell. But I don't use them all. I only use the stuff about mummies, or I only use the keratin. So I gotta find my edition that has mummies in it. Perfect. I found my gene. Now I've got that information. Like Lisa said, we better hope that it has stuff about mummies. I got lucky, we'll say this time. It did have stuff about mummies. I found that information. I'm really excited. I can write my book report, except Encyclopedia Britannica is too important to be able to leave the library. Like we got to make sure that no water gets spilled on it, that no dogs chew it up. Encyclopedia Britannica has to stay in the library. just like your DNA has to stay in the nucleus, because we don't want your DNA getting chewed up either, right? So for me to be able to get that information about mummies from Encyclopedia Britannica with the book letter M, I'm going to have to use a copy machine. Now, the copy machines in my library weren't quite this old school, but I decided to go all in on the retro motif here. So we got a copy machine, maybe a little bit more advanced than this. Take my nickel over to the copy machine, drop it in, and make a copy of the information they have about mummies. Now I have the information in particular from that book that I want and that I need to write my book report. Those copies that I made from Encyclopedia Britannica can leave the library with me, just like RNA can leave the nucleus. I can bring those copies out of the nucleus or out of the library back to my house where I can turn them into a book report, or where your cell can turn them into proteins. So when we're thinking about why we need to do transcription, why we need to turn DNA into RNA, 2 reasons. Number 1, DNA can't leave. It's too important. We don't want it to get damaged. It's too big. That's the first reason that we have to make copies. I can't take Encyclopedia Britannica home. I'm not allowed to. The other reason, number 2, that we have to do transcription is because we don't want all the information for every kind of protein in the body. We just want the information we're looking for. Just like when I'm doing that book report about mummies, I don't want the information about aliens or about tornadoes. I just want the information about mummies. Transcription helps us to keep our DNA safe, and it helps us to only build the kinds of proteins that we actually want. Transcription in an analogy. Help me out in the chat. What page in your notes do you have this analogy on? Okay, I'm hearing page 5. Okay, so page 5 is where you'll find this analogy. You'll also find it in the guided lesson. I want to pause for a moment and allow you to try to fill in page 5. And if we've got questions about the analogy, let's go ahead and put them in the chat. See if we can help each other out with them. What questions do we have about the chat? Max, I see your question. That's related to the table that's before this analogy. Guess what? That table is going to be group work. So I'll give you a chance to chat that out with your group. Max is asking about where we find RNA. That's going to be one of our questions on the group work assignment today. But you are correct. There'd be more than one place that we can find it. It can either, just like we talked about, I could take my copies back to my house, kind of like a ribosome in the cell, but at the same time, I could carry my copies around with me. I could take them to school. They could be really anywhere. So RNA can either be being turned into a protein, or it can be floating around in the cytosol. Although I'll mention with RNA that because it's only single-stranded, it is not very stable. It falls apart really quickly. So it's more likely we use it and then it falls apart. We use it and lose it, as they say. Yeah, so Sharmila asked if the term cytoplasm would apply. There's a term that's slightly better for our class. What was the term that we used instead of cytoplasm? Because cytoplasm includes those organelles. What is the soup by itself called? Yeah, Jessica's right. The soup by itself is called cytosol. So we could say that the RNA is floating around in the cytosol, or we'd have it attached to those organelles that we talked about that help us to build those proteins. Remind me in the chat again, who builds proteins? As a wrap-up review question, what 2 organelles did we say in our cells help us to build proteins? Exactly. Yep, so we've got those small ones that are the ribosomes, and we've got those big ones, that is the rough ER. Any other questions before we bounce into our groups? All right. Well, I'm going to go ahead and end my recording, and then I'm going to get you that link for our group work.